years. Now she is a professor of developmental biology and director of Beckman Center at Stanford University. She has truly a remarkable career and publishing nearly 300 publications, was elected to National Academy of Sciences at very early on of her career, probably in the 30s, and was elected and received numerous awards, including just recently I heard the Dean's Medal at Stanford right before she came to China. And in terms of the science, I would say anyone would take pride that what Lucy has done. She essentially funded the field of study of Caulobacter bacteria, and many people use this system now as a system to study cell cycle, study developmental biology, and as Lucy will talk, I'm sure, why this system why this bacterial cell is so powerful and so illuminating in terms of the basic principles of biology, and we'll hear that. Among the, I think, most remarkable thing is many of her students and postdocs now become full professors, and their generation, their students are now taking on their next steps. So we are seeing the F1s and F2s of Lucy Shapiro's lab nowadays everywhere. To me personally, I read of Lucy's work when I was even a graduate student on system biology of bacteriophage, where Lucy's name and her husband's Harley McAdams' name first jumped into my view, and of course their work on system biology has been so influential since then. And that was before even system biology was actually coined later on. So long story short, we are extremely delighted that Lucy agreed to come here and give this keynote speech. And let's welcome Lucy.
Thank you so much. That was a very lovely introduction. And I'd like to thank the organizers for inviting me here. It's a pleasure and exciting being in China. This is my first visit to your very beautiful country. And after the meeting, we're going to do some sightseeing too. What I want to tell you about is work that we do on a simple bacterial cell. And before I get into my slides, let me explain why we started working on a bacterial cell. And this happened soon after I graduated with my PhD. And I was trained as a biochemist. But what I wanted to understand is not what happens to all the enzyme systems once you break open a cell and take out everything. What happens when you use genetics to figure out what a cell is doing, but rather, what is the in vivo biochemistry? What are all the parts in the cell doing, and how are all these parts integrated? And as our work went on, it became clear that was very, very important to be able to do high-throughput genomics, to go everywhere from the chromosome to what's produced and all the regulatory mechanisms that happen. And most importantly, and people forget about this, they happen in a three-dimensional cell. So what happens not just in the readout of the chromosome matters, but where you put your protein factories to make the cell an integrated system. And what I'm going to tell you about today, the story I'm going to tell you about today, deals with Caulobacter, which is the bug I work on. And Caulobacter has a stalk at one end. And I'm comparing the cell cycle of this bacterial cell to a stem cell, because it's absolutely the same principles. When a stem cell decides to go through an asymmetric division, you get asymmetric protein localization, cell intrinsic polarity, then the spindle is oriented in a specific way, and then upon division, you get cells of different fate. Now, if you look at the Caulobacter division pathway, you have a polarized chromosome, and then an asymmetric division that's absolutely controlled by the differential localization of signaling proteins at the two poles of the cell, giving you two cells with different cell fate. And what I'm going to do in this talk today is go through each of these pathways to show you what the wiring diagram is, essentially, of the cell to allow you to have an asymmetric cell division. First, let me tell you a bit about what we know about Caulobacter genomics. A very talented postdoc in the lab, Beat Kristen, showed that there are 480 essential genes out of the 3767 ORFs. The Caulobacter genome is about 4 megabases. To do the essential genome, he made about 430,000 unique insertions and sequenced them all. And we were able to tell which regions of the genome were essential. And we did this one in rich medium, all right? And he showed that there was a large number of essential genes, 8 essential small RNAs, although we have many, many more small RNAs that are not essential for viability, but 8 are. 402 essential regulatory regions. By the way, this was all done at 8 base pair resolution. So it gave us an enormous amount of information. But most interesting to me is that there are 90 essential intergenic regions that have no known signature. They're not regulatory. They are intergenic. They're not regulatory. They do not code for anything. And they're essential. As I get further in this talk, I'm also going to talk about methylation of the chromosome, and a group of these guys contain sites that are protected from methylation. So just store that away, and I'm going to come back to that again. So before I get into the regulatory circuitry, let me just give you a list of all the databases that we now have for the Caulobacter cell. We've done 5' RNA mapping and identified the complete genomic transcriptional landscape right through the cell cycle, including both sense and antisense transcripts. And interestingly, the antisense transcripts, a group of them are also cell cycle controlled, and they control the transcription of key regulatory proteins. We've done 3' end RNA profiling and mapped over 5,000 transcriptional termination sites across the genome, discriminating between row and row independent termination, in collaboration with Jonathan Weissman, a postdoc in my lab named Jared Schrader, has just completed the ribosome profiling, moving right through the cell cycle. And I might add that this ribosome profiling turned out to be extremely important to get a very accurate picture of the annotation of the chromosome. And by putting together our mapping and ribosome profiling, we really now know where every single gene begins and ends. And the accuracy actually was pretty poor before we finished that. I will tell you about today the methylome, working with PacBio. we determine the cell cycle control DNA methylation state of the chromosome, which turns out to be critical for the cell cycle control. I just told you about the essential genome, and we've also done proteome profiling and identified 66% of all the predicted proteins. So now, with that background, and I'm going to refer to these as I go through this talk, let's turn to the Caulobacter cell cycle. That indicates the chromosome. In this swarmer cell, the chromosome is quiescent, and there is a single chromosome. Thank you. And you can't initiate DNA replication until you differentiate into a stalk cell. And by doing that, this flagellum is ejected, these pili are sort of sucked back up like a straw, and you then build a stalk. When that happens, only then can you initiate DNA replication. And then as the chromosome is replicating, you're turning on blocks of genes that code for the proteins involved in cell division, flagellar biogenesis, building a new flagellum here, pili biogenesis, and most importantly, the turn on of a gene that methylates a specific GANTC site on the chromosome, and it's adenine-based methylation. So what I'm going to first talk about now is the polarized chromosome and spindle orientation. So down here is the Caulobacter cell. It's a swarmer cell. Here is the old origin. When the cell drops this flagellum and begins to differentiate, the replosome forms around the origin, and as soon as the origin and the PAR-S centromere are copied, it moves very rapidly to the other end of the cell. And you can see it here. This is just using an algorithm designed by Patrick McGrath, a physicist who is in the lab of Harley McAdams. He was able to follow the trajectory of each locus that we looked at as you move through the cell cycle. And, for years, it was in all the textbooks that bacterial chromosomes segregate by attaching to the cell wall, and as the cell grows, they move apart. Well, this is simply not true. The movement of replicated parts of the chromosome occur before any change in cell length. And they may be linked later, but that is not the fundamental property. So then the big question is, we identified a sequence, which we call PAR-S, as the centromere, and that this PAR-S near the origin moves to the other pole and gets anchored. So what anchors it? Well, all of the guys who did this work is covered by this number. So I'm just going to have to tell you who they are. This work is done by Grant Bowman, a great postdoctoral fellow who's now running his own lab at the University of Wyoming. And Grant asked that question. What grabs the stuff at the cell pole? So here's a Caulobacter cell. And what Grant discovered was a protein here that he called PopZ that forms at this pole of the cell. And this is an amazing protein. It's a 19 kilodalton protein, a little guy, and it forms this huge network at this pole of the cell and is able to bind many different proteins. I'm not going to tell you all about this now, except that we've divided the protein up and we know exactly which portions of this protein bind to which proteins. Most importantly, it binds to the complex of this centromere, PAR-S, bound to a partition protein called PAR-B. So here PAR-B is in green, the PAR-S is in blue, and this is the PopZ complex. And if we now follow this during the cell cycle, and this is a time lapse of PAR-B labeled with CFP and PopZ labeled with YFP, we can see here that PopZ is at this pole, and then when you duplicate the PAR-S complex bound to PAR-B, it moves across the cell, and then the PopZ assembles at this other pole. And it's the initiation of DNA replication that signals the assembly of this PopZ protein at that pole. And we don't know that mechanism. We don't know how the cell is picking up that signal, but it does, and it grabs it there. If you get rid of PopZ genetically, then this guy, this newly replicated centromere, keeps trying to attach to the pole, and it can't do it. It tries and backs away, tries and backs away, and ultimately you can't complete the cell cycle. But if you now diagrammatically go through the cell cycle, here I show PopZ in red, the PAR-B attached to PAR-S in yellow, and here it separates. The initiation of replication occurs. New proteins then are built at this pole to assemble a stalk. And as the replicating centromere moves to the new pole, it's grabbed and held there, and then you're ready to start again. So we have very specific polarity, and how might this work? And now this is work of another postdoc called Jared Tassin, who showed that it is likely that this PAR-B, which now here is in red, moves directionally to the other cell pole. This is another segregation protein in green called PAR-A, which forms polymers extending from one pole of the cell. And as PAR-B, bound to the centromere, is duplicated, it meets up with the end of PAR-A, and we have an ATP-dependent depolymerization, which pulls the PAR-A, which pulls the PAR-B centromere to the other pole, and this model is called a burnt bridge Brownian ratchet. And now, how do you get directionality? How do you get orientation? And you do that by having both PopZ to grab things and another protein called TIP-N, which was originally found by Christine Jacobs-Wagner and Patrick Vioyer, that binds this PAR-A protein, and then this grabs and makes a complex with PopZ, TIP-N, and PAR-B, and it holds it there. So in fact, we wondered, well, what does this PAR-A look like? When you just look in diffraction-limited fluorescent scenes, you see PAR-A as a cloud. And we can actually follow this cloud retracting as the PAR-B centromere chews it up and moves. But when we worked with Steve Lee and W.E. Murner at Stanford Chemistry, using super resolution, the PAR-A localizes to a narrow linear structure running down the center of the cell. And we've seen this now in hundreds of cells. So there is at least an accumulation in a central location of this PAR-A polymer showing essentially spindle orientation. And in fact, it's rather interesting that if you look at a eukaryotic cell, there are common principles between segregation. So here in a eukaryotic cell and here in Caulobacter, we have dynamic polymers, polar organizing centers, and centromeres pulled to the cell pole. And now we can now look at this in a more cohesive and logical way. So now if I just summarize what I've told you, here is a Caulobacter cell. This is the chromosome. Here is PAR-S. Here is the terminus. And I've just lit up one locus, a random locus. We have now followed the movement of 114 separate loci through the cell cycle. And I'm just showing you one. So when the swarmer cell loses its flagellum and is able to initiate DNA replication, it forms this replisome. Replication can initiate. We have PAR-A-driven segregation of PAR-S. We anchor PAR-S, the centromere, to the cell pole by PopZ. And clearly, replication and segregation of chromosomal DNA occur simultaneously. Interestingly, if we now follow this one locus, when the replisome moves through it, it then moves as in a mirror image to the other pole of the cell, forming this guy in the right relationship to the centromere as it is down here. So there is a collinearity between where a gene is on the chromosome and where it resides in the cell. And so you have the immediate restoration of the conserved arrangement and then cell separation, and you're off and running again. So one of the interesting things about looking at a cell cycle in a particular simple cell is to understand how everything is coordinated. And one of the big surprises to us was that the polarized chromosome along the long axis of the cell actually dictates the site of cell division. And this is the work of Martin Thanbichler, another postdoc in the lab who is running his own, who was in the lab, running his own lab at a Max Planck Institute in Germany, his own Caulobacter lab. And he discovered a protein called MIPZ. And that protein is required for the mid-cell placement of the division ring. So if this is the origin region down here, it duplicates and goes through. This red stuff here indicates where MIPZ is. And what Martin found is that if this is the origin region, these are the centromere sequences, and PopZ builds along this region. MIPZ binds to PAR-B, but forms a gradient away from where the origin is. So that if we look at the MIPZ YFP, you can see a gradient from each pole with the lowest concentration in the middle. In the same cell, you can see GFP PAR-B. And this gradient has its highest position at the origin regions and lowest at mid-cell. What's important is that the MIPZ PAR-B complex inhibits the polymerization of the cell division ring. The cell division ring is made up of a protein called PHITZ, and it inhibits that. So now if we look at a model, this is a simple cell. Here is PAR-B, here is MIPZ. These are a bunch of PHITZ monomers waiting to polymerize into the cell division ring. And as you duplicate this guy, you decorate the centromere with PAR-B, and MIPZ forms on it. It moves across the cell, and as soon as it hits this PHITZ here, PHITZ polymerizes at the site of lowest concentration of MIPZ. So by having PAR-B at both poles and using MIPZ, which inhibits the formation of the PHITZ ring, you're forcing the cell to make the cell division ring at the site of lowest concentration. I could show you a movie if it works, but it doesn't, so I'll just explain it to you. So this is PAR-B and this is PHITZ, and if I could show you the movie, you would see that PHITZ was moving, and as soon as it hits that, this goes, as soon as this hits that, it goes to the center. So this guy moves there, and this moves to the center. Next, and this is something that's fairly new, and to me is pretty remarkable, and that is that the actual replication of the single circular chromosome serves as a clock to time the expression of cell cycle transcription factors. So going back to this cell cycle that I showed you and all the functional modules that do their thing, we have found that there are 4 master transcription factors that control 200 cell cycle regulated genes. And this work was done by a graduate student, then graduate student in the lab, Mike Laub, in 2000, who did the first cell cycle microarray of a bacterial cell. And he showed to our great surprise, that the cell cycle is hardwired for turning on sequential groups of genes, and we know most of them now, as you move through the cell cycle, over 250 of them. Mike Laub is now a professor at MIT, again, still studying Caulobacter and doing just elegant work. So if we now look at these factors that I told you about that control the cell cycle, here we are moving through the cell cycle. Let's start with one of these transcription factors, which we call GCRA. It's blue. GCRA is turned on here. It then turns on CtrA, another transcription factor, which controls a whole bunch of genes. And CtrA then turns on this DNA methyltransferase, which turns out to be critical. Back. The critical question was, what starts this whole cascade? And the DNA A protein has to be made to turn on the GCRA protein, but we didn't know what turned this on. And now we're going to turn in to DNA methylation. But before I do that, let me just reiterate and remind you that the DNA methyltransferase is made out here, and the cell initiates replication on a chromosome that's in a fully methylated state. So if this is the chromosome, here's the origin, here's the DNA A gene, here's the CtrA gene. When you begin, before you start replicating, the chromosome is fully methylated on the adenine base of GANTC. And so it's methylated both on Watson and Crick. It's fully methylated. Turns out that DNA A, which is a transcription factor for a large number of genes, as well as controlling the initiation of DNA replication, can only be transcribed if its promoter is in a fully methylated state. So if it's the promoter is methylated on both Watson and Crick. Another critical transcription factor that controls 95 genes later in the cell cycle can only be transcribed when its promoter is in a hemimethylated state. So here we are, just at the beginning, everything's fully methylated. DNA A is transcribed, but CtrA cannot be transcribed. As you replicate the chromosome, and the bacterial chromosome is replicated bi-directionally, when the replication fork moves through the DNA A gene, it moves to hemimethylated. You get 2 copies of hemimethylated thing, and then the DNA A cannot be transcribed. The replication fork has not yet moved through CtrA, so it's not transcribed. However, when that replication fork meets up with CtrA and moves through it, then what happens is that you turn on transcription of CtrA. So as we have shown now with these 2 specific loci, that as you move through the chromosome, time-wise, you're turning on these critical transcription factors. And most recently, and this work on DNA A was done by another postdoc called Justine Collier, who's running a Caulobacter lab now in Lausanne. And what she has recently found that both FITZ and MIPZ, those 2 proteins I told you about, are also controlled by the methylation state of their promoters. And so how did we get to this all? This just shows you the complete regulatory circuit. DNA A, when the methylation sites are fully methylated, controls about 40 genes and allows the initiation of replication. DNA A, before it's gotten rid of by specific proteolysis, turns on the GCRA gene, another replication controller, another transcriptional controller. GCRA controls about 50 genes, and it then, in turn, turns on the expression of another global regulator, CtrA, and it does so here when this is in a hemimethylated form. GCRA controls about 95 genes, and when it's in the cell, it inhibits replication initiation. CtrA turns on that DNA methyltransferase right at the end. So then it, in turn, starts the cycle again by putting everything in a fully methylated state. What's interesting is that this replication, this transcription factor, about 50 genes are controlled by GCRA. About 30 of them are only controlled based on the methylation state of their promoter. So we now know that changes in DNA methylation state controls the timing of the entire master regulatory circuit. And now I'm going to tell you a little bit about how we did this. We did this with Pacific Biosciences. In just 1 week, PacBio was able to show that as the cell moves through the cell cycle, the DNA sequentially, base by base, moves from full to hemimethylation. It took us, I can't tell you how many months. I said many months. I should say many, many, many months to prove this for just 2 loci. We were able to give PacBio samples of DNA from Caulobacter at 5 times during the cell cycle, and they did the full genomic sequence of the chromosome at all 5 times and gave us the complete methylome, every single modified base in that genome, and they did it in just days, really. It was pretty amazing. What really interests me, what I'm really excited about, is that 26 sites on the genome are these GANTC sites, which are so critical, are protected from methylation, clearly opening up some new type of regulation. 6 of these are essential for viability. They're in intergenic regions. We don't know why. And this, to me, is going to open up a new set of regulatory phenomena. While doing this, they also identified the sequence signature, and we've identified the methylases for 2 new adenine methylases and 2 new cytosine methylases. So how did they do this? Well, most of you know about PacBio sequencing using RNA polymerase and the incorporation of labeled nucleotides, and the way in which they pick up a methylated or modified base is by looking at the interpulse duration, IPD. In other words, how long it takes for the polymerase to bring in the next nucleotide if you have a modified base right before it. So here's an example of a duration time if you have a methylated adenine or an unmethylated adenine. And what we then did was compare two databases. One is we as PacBio, not me, sequence the genome as you move through the cell cycle, all these different times. Here's the circular chromosome. The origin is down here now, and as I told you, you replicate bi-directionally. So this shows you that there's more DNA where you've just replicated at both ends, and this stuff has not yet been replicated. So if you move through the cell cycle, you can follow the movement of the replication fork. Now, what we're going to do is we're going to follow these GANTC sites, methylated on A's, top strand and bottom strand, as you move through the cell cycle. So this indicates hemimethylation. This is full methylation. So this is the stuff that was first replicated, but now we're not looking at replication, we're looking at methylation state. And as you do this, you can follow every single base as you move through the cell cycle, going from full methylation to hemimethylation, so that the GANTC methylation state progresses from fully methylated to hemimethylated as a function of the cell cycle and the movement of the replication fork. Oh, I must say that PacBio was so excited about us doing this. This is just for full disclosure. I'm now on the board of directors of PacBio, so I've done my full disclosure here. Let's turn to the architecture of the cell's regulatory system. We've shown that there are oscillating levels of a small number of master regulators that drive the cell cycle. Functions are turned on as modules. Modules are turned on just when they're needed, and the synchronization of the cell cycle engine to the cascade of cell cycle functions ensures reliable operation. For example, linked via DNA methylation state to chromosome replication. Transcriptional regulation is only a fraction of cell cycle control circuitry, and we have to be very careful about that. Proteolysis, localization of proteins, the translational machinery, the methylation state, all of these things are as important as the transcriptional regulation and all the hairballs we see with transcripts turning on and off under many conditions. You must put that information back into the living cell and understand all the moving parts at once, or you can be fooled by what's going on. The overall wiring diagram is selected for robustness of function, and regulation by DNA methylation state is an essential cell cycle regulatory mechanism, and it contributes to fitness by enhancing robustness. Now I'm going to tell you just one more story, and I'm going to be done. And I'm going to go back to this slide, because now what I'm going to turn to is asymmetric protein localization, giving you asymmetric cells. We have shown that differential localization of both signaling proteins and proteases is critical to give you an asymmetric division. So I kept telling you about CtrA, this transcription factor that controls 95 genes. CtrA is present in the swarmer cell, and when it's there, it silences replication initiation. In order to initiate DNA replication, you have to get rid of CtrA by a very specific proteolysis system. You stop proteolysis of CtrA, and then the transcriptional clock turns on CtrA again, and it's activated, and it turns on all of these modules. In the pre-divisional cell, a barrier forms before division. The inner membrane closes, and you have CtrA up here and none down here, so that upon division, you can initiate DNA replication. So now let's look at how this is controlled. CtrA has to be activated by phosphorylation to do all the jobs that it has to do. It gets its phosphate from a kinase, a histidine kinase, shown here, mediated by a phosphotransfer protein. So I've shown you where CtrA is. Interestingly, CckA, this kinase, does its thing when it's localized to one pole of the cell. So how is this integrated? How is the controlled proteolysis and the asymmetry of CckA function to give you an asymmetric division? So now let's look at a dynamically localized protease complex, ClipXP. CtrA comes into this wearing blender, and it gets chewed up. So if we look through the cell cycle, we found that this guy is localized at this specific time in the cell cycle to the cell pole, bringing CtrA and a factor with it, and CtrA gets chewed up. When it finishes, then it goes to the cell division plane, where we've recently shown, and this was done by a graduate student, Brandon Williams, that in fact the localization of this protease to the mid-cell chews up the divisome and allows you to divide the cell, and then once this barrier forms, it again localizes here, chews up CtrA here, but not here. So now how is this localized protease system related to the localized signaling protein, most importantly that kinase? this is probably my last data slide. It does so because this kinase, which is the one that activates CtrA by putting a phosphate on it, also phosphorylates another protein, which is called CPDR. When it's yellow, then it's in a phosphorylated state. When it is blue, it is active in the localization of this protease to the cell pole, so that the activity of this kinase both gives you functional CtrA and prevents you from getting rid of CtrA because it keeps this guy in a phosphorylated state. So once the protease localizes, this guy meets up with it and degrades it. And now let's put this all back into the cell. So remember, these little red dots are all CtrA. And this work was done by Antonio Iniesta as well as Patrick McGrath. And what we found is in the swarmer cell, you've got this guy in the phosphorylated form. Remember, the phosphorylated form cannot localize the protease, and therefore the protease can't work. Then we no longer have CPDR phosphorylated, and everything goes down to this pole, and you degrade CtrA, and you initiate DNA replication. Remember, CtrA blocks DNA replication. Then this guy, this kinase, localizes its activity to this cell pole, and all this guy becomes phosphorylated again in yellow. And if it's phosphorylated in yellow, you can't localize the protease, so you don't degrade CtrA. And now as you move through the cell cycle, when the barrier forms, this is trapped at this pole of the cell. That gives you the asymmetry. And this asymmetry then allows you to get rid of CtrA down here because this guy is in its blue form. It's not phosphorylated. And we're able to then go back to where we were and give you an asymmetric division. And in summary, what I've shown you is that asymmetric protein localization polarizes the spindle apparatus, polarized chromosome sets the division plane, and asymmetric localization of signaling proteins and proteases gives you cells of different cell fates. And for that, I have to thank a group of extraordinarily talented students and postdocs. I've mentioned most of them as I've gone through my talk. Whoops. This group of guys has already left the lab and are running their own Caulobacter labs. This group are still in the lab doing really beautiful work. And I have to say that a lot of this was made possible because we made an interdisciplinary lab. And we collaborate very closely with Professor McAdams, and his students are generally getting their degrees in physics and engineering. But what we've done is we've combined the labs so that we have a physicist sitting next to a geneticist, an electrical engineer sitting next to a biochemist or a cell biologist. And the result of that was to allow us to do the kinds of experiments, the kinds of analyses, the design of algorithms that we would never have been able to do in two separate ways. And most importantly is having these two groups of people side by side and talking to one another so that they can learn from one another. So with that, I would like to thank you very much.
It's a beautiful talk. And so I want to ask you this question is, so you have this beautiful DNA replication-driven cascade. So in the bacteria, are there any hints or available example, or are you looking to this one in eukaryotes?
In eukaryotic cells. Clearly, methylation is very important in eukaryotic cells. There's no question that CpG islands and their methylation state controlling transcription on and off is going to be important. Nobody has looked as the relationship between these methylated regions and the progression of the cell cycle.
Yes.
Unfortunately, the most logical place to do this in would be yeast. Yeast doesn't have methylation, doesn't need it. It's gotten around it some other way. That's at least true in Saccharomyces cerevisiae. Now, possibly they can do something like this in Pombe. But I should add, this whole story about this critical methylation is true for a large group of bacterial cells. And it's been done in the alpha proteobacteria and not the others. So this is all fairly new. And the fact that we can now get out the full bacterial methylome in days will change.
Okay. So a second question is regarding, is this, because now it's this 4 methylation status, it can be activated. So is there any conditions for the bacteria, because sometimes bacteria, when let's say in a certain environment, it may not.
You mean, let's say, stress or being hungry or what have you?
Can be
yes,
regulated?
yeah. So you can see differences. There's no question about that. But you don't change the core. So some of the genes that are controlled, but the ones I've shown you are all essential. So those are not going to change. But there are going to be instances. In fact, those protected methylation sites, they differ when you grow under different conditions. Not all of them, not the essential ones, but others. So I think, this is new data. And we're about to go into this further, but I think it's going to be very important.
Hi, Lucy. It's a great talk. I was wondering, maybe I have missed that point, but can you elaborate a little bit on how the conversion of the full methylated DNA to the semi-methylated DNA during DNA replication turn on one gene while silences another gene?
Yeah,
Is
so
the mechanism known?
Yeah, so the key to that is that the DNA methyltransferase is only present for a narrow window of the cell cycle. And what you're asking is, when something is in fully methylated state, why is it red as a promoter? And another one, when it's in a hemimethylated state, why is it red? What's the biochemistry? And so this work is just beginning. Justine Collier in Lausanne is carrying forward with this. We don't yet know what factors bind to the differential fully methylated or hemimethylated. This is being done right now.
So if these proteins involved in this cell cycle control, if it's not so abundant, so how the cells maintain this process so robust?
Could you? I didn't understand the question.
Can you repeat again?
Yeah, if the copy number of these proteins involved in this cell cycle is not so high, so how they can control this process so robust?
Can you translate?
You get it?
If the protein level is so high, how can the cell to control them so robust to use this mechanism?
Yeah, well, some are high and some are not high. So CtrA is present in thousands of molecules. But we have a very strong proteolysis system to get rid of it. So proteolysis is as key as differential transcription and differential translation. So we have to remember that that's a key event in clearing the cell of these proteins. Yeah, and yes, sorry, Harley just reminded me. It's not just when it's there or not there. These transcription factors have to be phosphorylated to be activated. So actually, the active proteins are controlled at multiple levels. It's controlled by the level of transcription. It's controlled by the level of phosphorylation. It's controlled by proteolysis. And it's controlled by subcellular positioning in the cell. So there are multiple layers of control. And that's what I meant when I said it ain't all transcription. There are many other levels of control that are critical. Yeah, Job.
Fascinating story. So I'm fascinated by the intergenic regions that are essential, obviously.
Yeah,
So
we are
do
too.
they have a special chromosomal localization?
Yeah, so actually, they seem to be placed around the chromosome, the essential ones. This is the circle here, here, and here. We're just started working on this, Job.
So
And what interests me, of course, are these involved in structure.
That's
Okay.
clearly my bias.
Yeah.
in this field, but
Yes,
I
my
was just
bias
wondering
too.
whether the sequence itself might be essential, but is its position?
Yeah, well, we're doing that. We're moving them.
Yeah, that's really. Another question is that if you, I imagine that the localization of these transcription factor genes must be very much concerned.
Okay, so we've moved them. Yes, we've moved
So
them.
what happens if you move
What
them?
happens is the cells don't die. They become sick, and they're confused. And the cells are of all different sizes. That's all I can tell you. It's like you're screwing up the integrated circuitry. And, of course, losing robustness. So if you move DNA A, those cells will not survive a competition with healthy cells.
Oh, that was great. What happens if you remove the methyltransferase?
Okay, so in the deletion of the methyltransferase in rich media, the cells drop dead. Okay, they can't do it. If you delete the methyltransferase in minimal medium, where the cells are growing very slowly, they don't die, they limp along, they make very long cells, they can't divide correctly, and they're ill. But it gave us a great thing to work with, because we could then do a lot of experiments with that.
This is a unicellular organism. Can you comment on how general this mechanism are?
Yeah, someone else asked that already.
As,
The answer is, it hasn't been done. I don't know.
do you observe any similarities to asymmetric division in other systems?
In the methylation,
In asymmetric division?
just what I told you. That's the closest. This is what I know. I don't know anything further.
Okay.
But I'm sure it will be followed up.
But this is a single-cell organism.
Yes.
Would be very different from a multicellular organism, right?
Yes and no. A multicellular organism is made up of single cells that have to talk to each other. And so what I've done here is shown you the regulatory circuitry that controls one cell. You go to multicellular organisms where you add another layer of talking to each other. You still have regulatory subsystems that have to work together. So our challenge now is to understand the regulatory subsystems that build a tissue or that build a multicellular organism. This is a paradigm for what you have to know about every cell in a multicellular organism.
That's great. Let's thank Lucy again.
Thank you very
Really,
much.
thank you.
You have a break.
So we have some, I think we have a 20 minutes break, and then we can come back for the next session. Thank you.